Yeah, so I want to talk about data management as it relates to imaging, although a lot of what I have to say here also applies to all kinds of data. But I've been involved in doing bioimage informatics for a while, and so this is the source of many of the examples that I might give here. So to get started, I want to consider what is the data flow in our imaging practice. So we might think about the flow going from the microscope to some sort of visualization, maybe making some measurements, and then going to publication. That might have been the actual workflow when I started. For example, we would actually put a photo camera on the microscope and take the roll of film to a dark room to develop the negative, and then look at that, at the photos. And if we needed to measure something, we would put a ruler to the photo and take some measurements there. And then everything would be written down, and the photos would be put with the paper for publication in a journal. So this is the workflow that basically kind of predates informatics applications to bioimaging. What we have more nowadays is something like this, where you acquire digital images that end up in some form of digital storage, some disk somewhere. And before you even look at them, you might need to do some computation. For example, in super resolution microscopy, you take tens of thousands of images from which you extract coordinates of events, localization events. And from that, you reconstitute an image, which is usually the one you look at. And so you might have actually several rounds of going from processing the data, looking at it, and do more processing. So for example, you might look at the data, do some annotations or select regions of interest before you do computation again. Obviously, at the end, you want to publish all this, so you would publish some description of your project in a journal, but the data would, especially the images, would be released to a repository like the bioimage archive, for example. So this is a lot more data movement in a way, and a lot more opportunities for things to go wrong. And what we have to keep in mind is that we want the data to be usable in the first place by ourselves, but then also by some close collaborators, but eventually after publication by other people. And reusable can take different forms. So it might be that you have a backup and you want to go back to the data, so you want to be able to retrieve that data. And if you come back to data, or you have no idea because it's someone else's data from, let's say, the bioimage archive, you want to have enough information to be able to know what it is about and how you can reuse it. So there are a number of things we want to avoid, and I have examples of all the of situations for all of these points. So for example, losing the data. And here, for example, we had the case, someone visiting the labs for several months as part of a collaboration, acquiring a lot of data. And the week before leaving, all the data are on the laptop of that person, and the laptop dies a few days before they left. And so there was actually no backup because the facilities had deleted the data because the user had actually taken the data with them. So from the facilities point of view, that was a user problem, but the user had basically no backup. So that's something you might want to avoid, especially if you're in the last phases of your PhD or postdoc. You also want to avoid that if someone asks for the data that you can't produce them. That's if you publish a paper, someone eventually might actually ask you for the underlying data. because they want to combine them with their own project, or they have some specific question they want to address on it. And that sometimes happen also at the review stage of the paper, where someone says, "Oh, I want to look at that data before I can actually review that paper." Also, you can imagine that you're following up on someone's experiments. Let's say you're starting a postdoc and you want to follow up on a previous student's work in the lab. So you want to have the same reagents. So you need to know what they have been using for those experiments. And also you want to be able to answer a question about the data that you have acquired. Someone might come along and ask you how this particular number came about. So you want to be able to answer that question by tracing basically all the process from the acquisition to the number, and say what happened to the data to arrive at that particular number. So where we are now about data management, I find that for a lot of people I've been talking to, we're in a transition period where actually many people say, well, what we've done so far always worked. So we haven't had any of the problems that I mentioned before. So why change? There are a number of things we want to change, reason we want to change, and some of them have to do in trying to produce more efficient and better quality research. And what we want to do in particular, increase the trust through transparency and reliability of the research process. We want also improve inclusiveness in the sense that whatever knowledge is produced is not limited to a few groups of people, like the old boys club type of network effect, so that everybody can actually access that knowledge, and to also do that, access the data. So that leads us to follow open science principles, and many funders, research institutes now adhere to those principles. So for this, you might also want to follow some good data management practices. And on the matter of trust, this is something that can come up. Which of these microscopy images do you think are real? I can leave you a few seconds to think and look before I give the answer. Well, all of them are actually fake. They were generated already a few years ago for this paper. And if you believe that paper, they're all deep learning generated images. And I'm saying if you believe that paper, because although they talk about how they generate fake images, they don't even actually provide any code where you can actually reproduce those things. So they might as well have taken sections of real images and said that they have been deep learning generated. So you see where we're going now. We can generate a huge amount of fake data. And now how do we trust that this data is genuine in a paper? Well, from my point of view, it's because you can reduce and increase the trust and reduce the doubts by providing an auditable trail from the data acquisition to whatever you publish. So here I would just want to mention something about FAIR, because you're often seeing open science and FAIR principle mentioned together. So the FAIR principles means findable, accessible, interoperable, and depending on where you look, either reusable or reproducible or both. But FAIR doesn't actually mean open in and by itself, but it's used to support open science by basically emphasizing the machine actionability of the data handling process. And that's due to the fact that we want to deal with high volume and complexity and speed of the data creation. That basically means we want to be able to retrieve the data and deal with it with a computational approach rather than having to dig through things manually and talk to people to find about the data. So this is about dealing with metadata and open formats for publication. And very often FAIR principles are mentioned at the publication stage. But what I want to say here is that the FAIR principles are also applicable during your project execution. You want the data to be findable by yourself, but also by your collaborators. And you want to ensure that everybody who needs to has access to that data. But you and your collaborators may need also to deal with the data with different types of software or environment. And so the data needs also to be interoperable. And it needs to be reusable, reproducible in the sense that whatever you do to the data, someone among your collaborators should be able to follow and reproduce or reach the same conclusions or derive data from the original. So FAIR principles are usually mentioned at the publication stage, but you should keep them in mind during the execution of your project, and that would be very helpful at the later stage. So what do we need from a high level perspective? We want to have basically a data management policy and also a platform that is a set of tools that helps us manage the life cycle of the data. So I mentioned all these data flow already from the original slides, but we want to have actually the tools and the policy to deal with this. And in particular, we want to identify and track where the data is, how the data sets relate to each other, and how things are being processed. And so what we are doing to the data, we want to document that. We want to also associate reagents to data or parts of data. For example, typically we want to know what is in a particular image channel. And by that, I mean, it's useless to tell that channel 1 is GFP because we don't know what actually GFP is showing us. We want actually to know the biological entities that we are looking at. We want also to facilitate data processing and in particular avoid or minimize data movement, especially for very large and complex data sets. And of course, we want to use standards for all of this so that we actually ensure interoperability. So here I want to show you, in terms of platform, what we actually planning at EMBL. So this is our roadmap where the data flows from the instruments on the left here. This is basically a bunch of microscopes. And what we want to do here is convert the data to OMISAR and store it in an S3 bucket. So we run our own S3 compatible storage here, but it could be anywhere, doesn't matter. And as part of that conversion process, what we would do is store the original data, so the one coming out from the instrument, in a tape archive, so that we can come back to it eventually later. And this process, we want to actually drive from a laboratory information management system that captures all the metadata. So that basically has the information, the metadata about the images, what instrument was used, but also what samples were there, linked to whatever is in the bucket. So and from there, we could eventually publish the data to the bioimage archive. But also we access the data through our computational processes, so various services. And here Typically, we want to use workflow management systems for dealing with the data, because that allows us to record properly what is done to the data. And I'll come back to this later. And here, our preferred workflow management system is Nextflow, at least for images. But of course, dealing with images is also as interactive steps. And for that, we also have virtual desktop that allows to use graphical user interfaces, and also sometimes web applications, and all that connected to this S3 cloud-based storage. And everything can exist independent of the backend, because we are leveraging cloud technologies. And eventually, we want to publish our workflows also on Workflow Hub, which is a registry of computational workflows. So what do the metadata do? I mean, if you think about being in a kitchen and you're looking for some oil for preparing your dish, and you have a bunch of bottles, of course, you could look at the color and make an educated guess about what the bottle of oil is. But in principle, you want to rely on some label. And that's basically the metadata we have for our images. And in this case, in the kitchen, the metadata is directly kept with the data in the form of a sticker on the bottle. So how do we keep and find metadata with images? And there are basically three non-mutually exclusive ways. And I'm talking on this part, the relationship between the storage and eventually the laboratory information management system. So you can keep the metadata in the names of your files and maybe directory names, or basically also encode some relationships in the directory structure. And you can supplement this by having companion files, so extra files, preferably structured rather than just free text, that you keep somewhere alongside the images. But you might also want to put things, or you might have part of the information in a laboratory information management system. So basically in a database somewhere. But that thing needs also to be able to reference the images that are linked to whatever information you have in that database. So if that's not the case, you need to be able to bridge in some form the link, to find that link. So here is an example of a project structure on the file system that actually encodes some of the relationships between things. So you have an overall project directory, and you can subdivide it with data, workflow, output, documentation. And then inside, for example, the data, you can differentiate between the primary data, maybe something that comes out of the microscope, and have maybe different experiments underneath that. And here I have listed a metadata.toml file, which is basically one of these companion files using the toml format, which is just a plain text file that can contain key value pairs, for example, of information. You might want to put final results like data tables of numbers into the output things, and you could put it under, like, for example, these feature tables as shown on the right-hand side. You will find an example or a template of this under this Git repository here. But you can feel free to devise your own. The main thing is, if you devise it, then you should also be clear about what goes where, and that's part of your data management plan. Also, the naming should be defined there. So I have a few rules for how to deal with all these things. So the first one is that you want to make sure that things are machine actionable and interoperable, because that's basically everything is processed by computers or programs on computers. And so for that, you want names that are unique, consistent, somewhat indicative of content, because you are also looking at it and you want to have some information when you look at file names. And you also want this to be compatible with various systems. And here I'm mentioning that because, for example, you can't use all characters for naming S3 buckets or files in X3 buckets. So there is a limited number of characters you want to use. Also, very often, white spaces are used as separator characters. So please don't use white space in file names, because this makes it difficult and is prone to errors, because the two parts will be seen as different things. Now, to be a bit more precise, I have these rules for file organization. So it's all kind of, I think, common sense, but you need to think about it. You want to define the directory structure that I mentioned before, and the content of the directories and the data files, so what goes where. You define naming conventions following some of the rules I mentioned before. You want to stick to some file formats for the various data types, and so not have a mixture of data types for, let's say, your images. So you don't want to have images in TIFF and some of them in PNG and some of them in whatever proprietary format, because then it makes, again, downstream processing difficult. So we need to be able to assume everything of a particular type is in a particular format. So this goes along the line of being consistent with everything to facilitate automatic processing. And ideally, you want to store some documentation with the data, maybe in the form of README files, maybe in the form of structured metadata file. And finally, something that you might want to consider is also to define version control procedures, because sometimes you update your data, you might run the experiment a different way than the first time around. And so you might want to be able to differentiate what is data that has been produced in the first round from data that has been produced in the second round, and again, for the derived data that are coming from the different rounds. Here is something about spreadsheet, because this is my pet peeve. I want to ban Excel. I wish that scientists had never heard about Excel, because basically nobody really knows how to use it properly. It was never meant to be used for science purposes. And there's been a number of papers already publishing every few years about how Excel mangles gene names, but automatically converting gene names to dates, if they even look like a date name. But it also happens that they convert numbers to some other formats and to some other things if you're not careful. So I ask my collaborators to always give me plain text tab delimited files. And that also avoids things like, oh yeah, the data to be processed is in the green cells, and the failed quality controls are those in red. Good luck asking my script to distinguish green and red in Excel cells. Might be possible, but probably not very reliable. And again, Excel is not really the most interoperable format for various reasons. So you want to have plain text files, you want to have row and column headers that are also unique and unambiguous. And you want basically each row contains as much of the data or the relevant data as possible. And you don't want to mix primary and derived data in the same file. So if you have derived data, you don't create another table below the first one, or you don't append to the original one, you just create another file. Also, the meaning of empty cells should always be to be unknown or undefined. If you leave a cell empty, that's what it means. It doesn't mean 0, it doesn't mean some other number or something else. And you don't put more than one type of data or item in a cell, so no concatenations of multiple things into one cell. Just create more columns if you need to. And that's important in particular when you deal with image derived data like features of segmented cells or other objects. Now, there are more things that you want to capture in a bioimaging data flow, and that's relationship between different things, and especially when we start having more complicated workflows. So let's look, for example, at a light and electron microscopy and correlative light and electron microscopy experiment. You start from some labeled sample, fluorescently labeled sample. You go through light microscopy imaging, so fluorescent microscopy. You get some image files. From those, you define some target regions that you want to look at by electron microscopy. So the same sample now is also looked at through electron microscopy, and you generate data for some regions of interest. So a subset of the regions from the original light microscopy file, and maybe from those you actually compute some more information. So you have derived data sets. And so you can capture these relationships in different ways. One way we do it is basically we know that we have a sample that goes through light microscopy, and we capture the relationship from the correlative experiment by actually making the electron microscopy derived or linked to the light microscopy experiments. And then now we can go from the region of interest that we have defined in both cases, they are linked back to the original sample. But there are other cases, for example, multi-omics experiments. Now, with case where we do the same thing, we have a labeled sample, we take a fluorescent microscopy image of that, and then we dissociate, let's say, the cells. We go through a cell sorter and we do single cell sequencing. And so on the one hand, from that sample, we have image files, and on the other hand, we have sequencing files, like maybe FASTQ files. So we want to also keep track of the relationships that, in particular, the fact that we have FASTQ files from the same sample from which we have acquired fluorescent images. So for this, the best tool for the job is a relational database management system. And I know a lot of people are reluctant to implement that because they see it as a lot of overhead. But it's really helpful to have one. And in particular, because the relationships are everywhere, not just where the few examples I've shown, but even deeper inside some of those aspects. So you can try to answer a few of the questions here, and generally, if you answer yes to at least two of those questions, you probably want a relationship, a relational database to take care of your data. So in particular, when you start having to track information across multiple files, if people need simultaneous access to the data, maybe changing the data, you want to manage basically the synchronicity, the access to this. You want to avoid multiple people changing the data at the same time and things like this. So far, I've talked about this, mostly about this left part of the roadmap. So what we do with images, a little bit about the derived data, but mostly from the point of view of storing information and tracking it in storage. Now I just want to spend a few minutes on the other part, which is basically the data processing and data handling side of things. So if we go back to the kitchen analogy, you have some data that you want to process to generate a result. So the data will be your ingredients, and the result, the output, will be this cake. But having the list of ingredients, however detailed it is just not enough to make the cake. What you actually need is a recipe that tells you in which order, for example, you're going to mix the ingredients, but also various things such as which temperature you need to do to cook things, and maybe you need to do things in different steps, in multiple steps, and all this is recorded typically in this recipe file. And this recipe file is equivalent to a computational workflow. So when you want to generate derived data, let's say in this example, you want to have a table of measurements about the cells or the nuclei that are shown in the images on the left. This is done by following this computational workflow. And that having it written down and as a program essentially means that it is almost self-documenting. So I want to go a little bit more into workflow. So computational workflow is a set of tasks, and they have dependency. You can specify the order of the tasks to be executed, and one task output can be the input for the next one. And each task needs also computational resources that you might want to specify. And then there are metadata. Metadata like the input data, but also the code that is executed, maybe also the version of the software in that case, and maybe the parameters that you're using for various algorithms. But there are also like the timestamps, maybe some exit status, like did it error on something, the amount of resources used, and so on. And then also you need to have some data management functionalities in your workflow. You need to find the inputs, and you also need to send the outputs to the right place, and maybe save some of those metadata I mentioned just above. So how do you document a workflow? Of course, traditionally, it has been about writing steps in a lab book. This is error-prone, inaccurate most of the time, so that makes things basically impossible to reproduce. So you could have scripts, but scripts takes some extra effort to properly document. So computational notebooks go a step further, because you can actually have let's say the plots or whatever derived output shown directly in it, but still it requires some effort to properly document a number of metadata. So really the best way is to use a workflow management system. And that's basically what I mentioned at the beginning. That's what we're trying to do. So we might use manual steps and interactive tools to set up the parameters. So maybe we look at Fiji for quality controls, or we use it to actually test a few things in terms of finding the right parameters for thresholding, for example. But everything else then from now on is run in an automatic manner using Nextflow. So there are good reasons for using the workflow management system, because they do things that your scripts probably don't do unless you actually code them yourself. So I mentioned the data management. Typically, the workflow you will tell the workflow, and it will be recorded in the workflow where the data comes from and where it goes. But also internally, it will track the provenance of the data and some of the metadata. You can also run and monitor those jobs, and also typically the workflow management system will allow you to retry failed jobs, so you don't have to run everything again if something fails. It allows you to manage the scheduling and the parallelization, resource provisioning. So this is nice features when you run on a compute cluster. You can have packaged software, and you can tell which version, for example, in which container you want, and so it tracks very precisely what is the software you're using, and it provides some portability in the sense that, in principle, it can run in many places under different environments. But For specifying workflows, you have to use a specific language, a so-called domain-specific language, which is the formal way of specifying the workflow. And it's similar to scripting. Here on the left, I'm showing a small example from SnakeMake. On the right, something from Nextflow. But you also have domain-specific languages that are not necessarily associated with an execution engine. So you could also use something just to describe the workflow, but not necessarily for actually running it. But of course, if you have an execution engine linked to it, it also makes running things much easier. And finally, I just want to quickly mention how to publish everything. And I want to mention this Crate initiative, which was basically a way of publishing or packaging a research project and the whole of it as one, so that everything is together, or at least it's linked all together, so that you can actually properly document or recreate the whole thing. And so basically, you might want to see this as the future of publishing research. And the research paper is really just paid advertising for your work. The real stuff will be publishing your data and your workflow, computational workflow, in a way that people can actually trace and audit whatever you've done, but also apply it eventually to their own research project. And I just want to finish with this data management checklist. And again, it's not unique to imaging, and you can access it with the slides later, so I just won't go through it. But I think this is where you should start from. And with this, I want to conclude. And if you have any questions, I'll be happy to answer them.
Very good. Thank you very much, Jean-Karim, for a wonderful webinar. And thanks, everyone, for joining in today. So we have 2 questions already, which are open. But while we are taking these questions, feel free to type in your questions if you have. So I can read the first questions now. So how could we identify so in the beginning of your presentation, you showed a image and asked the audience that which one is generated by AI. So one question is, how could we identify the image, whether it's generated either by AI or not?
Yeah, and this is basically because you would have an auditable trail that basically will enable you to trace the images all the way back to the regions that have been used and the instruments and every steps of data processing in between. So it might be possible in the end to fake that the microscope metadata, the samples metadata, but eventually it's traceable to physical things. So you would have to demonstrate that it's traceable to an existing microscope with actually settings that make sense for that particular microscope, and that the data processing, the samples also exist, so that you are able to produce the cell line that has been used in the image, for example. Or you can track the sample to a particular biobank sample, and so on. So having an auditable trail is definitely something that would limit the use of fake data. Because fake data, if you wanted to make it really look like the real stuff, you will also have to fake all the metadata and the research object inputs. So like the original microscopes and so on. Because you will have to say, well, this image has been acquired using this particular biobank sample provided by this particular lab and acquired on this particular microscope in this particular facility using those particular settings. Now, if those things are not consistent, then the data is probably fake.
Great. Okay, thank you. Now, the next question is, what what do you do when you're dealing with lots of images or basically big data? So how do you efficiently link images to experimental metadata in a LIMS system? For example, for each image in a screening experiment, there are several metadata categories: cell type, medium, culture time point, drug treatment, concentration, etc. that should be bidirectionally linked to the images. How should this data be captured and linked efficiently for thousands of experimental conditions and tens of thousands of images?
Yeah. So I've been doing a lot of screening projects over the years. And I always found that the best way to deal with the data is to have a database. And otherwise, you have your data split across multiple files. And typically, people start with various Excel spreadsheets, so which become quickly not manageable. So you have a spreadsheet that describes the layout of the plates, so what goes in which well, but only for, let's say, if you do a compound screen, so you put in one spreadsheet whatever goes into each well of each plate, but then you have another spreadsheet that puts basically similarly what goes in each well in terms of maybe the sample, if you're looking at different cell lines or different genetic backgrounds, for example, and so on and so forth. So already you should be combining those two tables into one. Then for screening projects, it's actually done sometimes by the microscope software. There is actually some naming conventions where and file system structure. Typically, you organize things by and the software does that sometimes for you by plate, and inside each plate directory you have maybe another directory for each well, and so on and so forth. But sometimes not. I mean, I've come across a screening software that organizes everything by time. And so if you run an experiment overnight, it will split your data into two different folders, one for yesterday and one for today. And now you have a hard time figuring this out if you don't use the vendor's software. So be careful about this. But again, you should have a data management plan first to decide what you're going to do. But again, if you want the best thing, it's a relational database.
Great, thank you. Okay, so the next one is for fair bioimage sharing. Providing the raw images with all relevant metadata might still not be enough to make them easily accessible to other scientists. Many imaging modalities require post-processing, for example, 3D reconstruction of light field data or spectral unmixing to make it meaningful. So how do we share raw versus processed images, or both, or backup, and how to grant access
So
this data?
yeah, there is quite a bit to unpack here. First, I avoid the term raw data because, as alluded to in the question, there are many steps very often. And so I usually talk about primary data. And the primary data is where is the starting point of the analysis that deals with the biological question. So there would be some community accepted primary data. So some pre-processing steps from the moment the thing comes from the captor, the camera, to the moment you start your analysis. Those steps might be considered standard. And at that point, if they are reasonably well accepted, then I would not necessarily bother with those. But the primary data would be the point from which you start your the analysis that addresses the biological question that you're interested in. So, for example, in super resolution microscopy, again, we acquire tens of thousands of images, and we record all those blinking events. But really, the post-processing is about identifying the precise localization of each individual fluorophore, regardless of how many times they blink. And so the primary data in that case is the set of X, Y, Z coordinates plus maybe some extra parameters depending on the method used. And that's the primary data. From there, either you directly process or analyze that data, or you reconstitute or regenerate an image, which is the one you use to address the question, the biological question. So all these tens of thousands of images that you have acquired before that, they might be of interest for someone working on the method development to find those XYZ coordinates of fluorophores. But otherwise, to address the biological questions, they are not particularly relevant because you're starting from these sets of coordinates to start addressing your question. So I think it will be partly community-based and depend on what you envision for the downstream use of the data. Of course, the more you provide, the better. But then in that case, if you provide in super resolution microscopy, for example, you provide all these original images, so 10,000 or 60,000 images, you will then also have to document how you go from those to the XYZ coordinates so that people can actually say, well, I want to try something else and see if I get the same result or something else. So you have to think, but I'm not thinking in terms of raw data, but in terms of primary data. And the primary data is the starting point of the analysis that addresses the biological question of interest.
Okay, thank you. Okay, so the next one is about S3 bucket. Efficient S3 bucket access control for different users or groups is not easy, especially for non-AWS ones. Do you have any experience to share in that regard?
Yeah, actually, I'm not sure it's not easy. It's maybe if you don't really take the time to learn how to do it. But we do this here for several projects. We have actually our own S3 storage, and S3 is actually an API defined by AWS. So when we talk about S3, we're actually talking about how to access the data using the S3 API. So it became a de facto standard. So when you see S3 mentioned, there is at least the assumption that it is compatible with the AWS API. And most non-AWS buckets, so self-hosted buckets, they are run using this Minio implementation, which is actually tested to be largely compatible with the AWS API. So this being said, it's a slightly different ways of dealing with things because it's not a file system, it's a web thing, it's a cloud thing. So, but you just need to do the step to learn how to do it. But also as the tools mature, especially for image analysis, it should become also easier and more transparent for the user eventually.
Great, thank you. Okay, encoding metadata in folder names and structure is common, but risks losing metadata when sharing or moving parts of a dataset. And once taken out of context, individual images lose their metadata. How do you mitigate this when using folder structure-based metadata? Is it preferable to write this metadata into individual image files instead?
So you can do that. So this would be the best way of not losing the link between the metadata and the image itself. So this is basically the idea behind OME-TIFF, where you use the TIFF header to encode OME structured metadata. But when you do this, you have to be also whether you choose that or you choose to do some companion file, as I mentioned, basically a metadata.txt file or something that goes in the same folder as the image, you still need to be disciplined enough to actually provide the information. So of course, when you move things around, you risk losing things. But most of the time, things are lost by just not being recorded in the first place at every step. So when you acquire the data, it's relatively easy to capture instruments metadata, because if you ask the microscopy software to do that, probably most of them will do. But this is metadata that is used for or necessary for limited number of use cases. What I want to know, and I've tried to run several projects over the years about reusing published image data, and they all failed because there is not enough experimental metadata available, including in the paper that has published those data sets. So you really want to have a system that also enforces the capture of the metadata. And that's why we really want to have a relational database here, because that constrains the things, but also what we've been floating at some point is that people could not book a microscope in the facility if they haven't actually filled the experimental metadata for the microscopy session that they are going to run. Of course, there is a lot of user resistance to that, but in the end, you have to be disciplined enough. Also, encoding data in file names and file structure requires quite a lot of discipline. In my experience, nobody is able to write the same information twice in the same way. So I've had basically people put metadata in the file names, but they use different orders on different days. So sometimes they start with the cell line, and sometimes they start with the date. And so that's cell line name, date, whatever experimental ID or something. And sometimes they start with the date, then the experiment ID, then the cell line name. And then they make some capital letters and small letters. And so you might have protein X with a capital X, and sometimes protein underscore X with a small x. So you want also those names to be generated by scripts so that it's consistent. And so typically, again, this is much better for high throughput screening because this is done by the software, the acquisition software. But when you do kind of manual low throughput experiments, that's where most of the problems are. So I'm not too worried about high throughput stuff. It's a lot of data, but because it's a lot of data, people already kind of anticipate things. And so it's usually done at least with a little bit of foresight, a little bit more discipline.
Great, thank you. So the next one is regarding AuroCrate. I guess it's AuroCrate.
Yeah.
Is it possible to directly deposit the data onto a repository, for example, Dataverse or something else?
So, yeah, I haven't talked, for example, to the bioimage archive, but maybe I would assume that biostudies would take our crates. Although you have to maybe talk to them because this is still not a very common thing to do. You can always put it in some data dump like Dryad, Zenodo, or something, because it will give you an identifier. The point of the crate is that it brings together everything, and so not necessarily all the data is in it, but the links to the data and to information. You can have a look at the documentation page or the project page. And if you're really interested, they have regular meetings. I can't remember if it's bi-weekly or monthly. There's a community there that is really helpful if you want.
Great, thank you. And we also have a webinar in this series planned. It's from Bioimage Archive and Biosamples. So these are public repositories. So I think you can ask this question again there as well, just to get more information. Okay, thank you. And the next question is asking for a suggestion. Is there any free or open source software that you can suggest to start a lab database?
Yeah, so we have actually our own system here, which is called LabID, and that has recently been made public. You would have to look up LabID EMBL. I don't have I think it's in gitlab.com. And you can basically find this. And what it does, it's basically registering all these relations, samples, and stuff like so. It's a full laboratory information management system, but it also has electronic notebook capability built in also. And then, of course, it's developed here. So if you have questions, you can ask the developers. I think you can raise a GitLab issue on the project repo.
Great, thank you. I've shared the link for LabID in the chat, and I'll also share this in my email that I'll send by tomorrow to everyone. Okay, so next one is any specific database technologies you would recommend for image related project?
So if you really only care about imaging and not so much about metadata, I should mention Omero, which basically is an image database. Omero, but Omero doesn't have really full support for all the stuff I mentioned. So it's good for managing some images and have some metadata in there, but it doesn't necessarily go the full way as LabID, for example. Otherwise, in terms of raw underlying technology, if you don't want to go with a server, I would recommend going with SQLite. It's basically a single file relational database, a very powerful, just a file a single database file, and so you can easily move it around if you need to. And of course, you can have PostgreSQL, MySQL type of setup, but those require to set up a server. Otherwise, you can use SQLite. It's robust, battle tested, can ingest large amount of data and everything into one file.
Great, thank you. So you mentioned the relational database, and the next question is about that. I'm curious about the relational databases. Never thought about using them so far, but I agree the advantages are immense. How to get started with that? I know only SQL so far, but I think it is for tabular data. What relational database is good one to get started for images?
Yeah. So what I mentioned before, so relational databases are all about tables. So you put things in tables. So you have to design what is called a schema, which is basically the set of tables that go into the relational database. And so as I mentioned, a very good way to get a lightweight way of getting started is to use SQLite. Otherwise, you can get started with either PostgreSQL or MySQL. What you should, so SQL, I want to say SQL stands for Standard Query Language. So it's a reasonable standard. It has been a standard for like decades, more than 50 years, basically. And although there are slight implementation differences, an SQL query is more or less standard across the engine. So as long as you don't use these engine-specific additions. So if you know SQL, I don't know what you mean by that. If you know the query language, then you know how to query things. So the trick is actually about designing the database schema. So basically the set of tables and what they should contain that goes in there. So of course you can have a database with a single table where you put everything. And for example, SQLite has a way of ingesting CSV files, for example. there are two things that I want to say that you should look into. One is about so-called normal forms, and you want ideally to have your database up to third normal form. So you can take any book on databases. For example, a good introduction used to be the MySQL book, because it started. I don't know which edition it is now. I used to have an old one, but it walks you through some of the steps. So you want your database into third normal form, which basically means that you don't repeat data. So you have a piece of information in only one table and one cell of one table and things like this. And the other one is you want to set up indexes. So the power of a database is that it's fast at retrieving data, even across multiple tables. But for doing its job, it needs to use indexes. And so you have to know which table columns you're going to use, and you have to put indexes on those. So again, I would say look at tutorials on database design. The books for MySQL and some of the others are actually quite good, at least the first few chapters before they go into specifics. Otherwise, I'm sure there are other things online available. I can look up a few of the more recent things if that helps. They will be put in maybe in the email that's circulated.
Great, thanks a lot. So we are already 6 minutes over time, but I think I can probably take one last question, if that's fine with you, Shankar.
Yeah, sure.
One, when using databases, how do you deal with data life cycle? That is, how do you ensure that links and locations remain intact when moving data from hot to cold or to glacier storage, for example?
Yeah. So your database should record what happens to the data. So when it's put on tape and deleted, that should be recorded somewhere. Typically, we put things on tape, and sometimes it's not deleted. So we know that we record that it has been archived and it's still there. And then if it's gone from its place, we record that it's, if it's archived and deleted, we record that. But if it's gone, we just record it's gone. Typically, we also avoid this partly by actually having the management system own the data. So it becomes read-only for the user. So once it's captured by the system, the best way to avoid issues is to have it owned by the management platform, so that it's readable by the users, but it can't be moved without going through the management system, so that the operation of moving the data or deleting it is recorded.
Great, thank you. Sorry for running over time, but there was just one comment. It's a question which came in about the primary or raw data. So thanks, John Karim. Good point with the primary data. One thought was maybe that someone might want to use different algorithm to process raw or primary data, whatever you would like to call it. In some years, maybe other algorithms become available and the raw data should still be backed up if someone wants to try to analyze it again.
Yeah, so I alluded a little bit to this point, and I think it should be, again, a bit community specific and how things happen. So that also touches on the life cycle of the data. How long do you want to keep the data available? Because technology evolves, and some of the older images can be acquired again with much better technology nowadays, because they are using readily available samples. So for example, standard cell lines that you just need to get from the freezer. Of course, if something has been generated from very precious samples that are not easy to obtain again, you might want to keep that for a longer time, and also maybe keep more of the upstream data. But eventually, everything kind of becomes obsolete. And then so far, even at the repository level, the policy has been we keep everything forever. But I don't think this is a fully viable strategy because eventually the usefulness of data decays. Although you never know. I mean, the MitoCheck data was generated 20 years ago. And up until actually a year or two ago, I was still getting one request every year for the data. So you never know. I mean, even if it's old technology, it might still be useful to someone because it's still images and they might want to still use it in some ways.
Thank you very much, John Karim, for the wonderful talk today and sharing your insights with our attendees. And thanks everyone for joining in today. I would highly recommend you to register for all the upcoming webinars which are part of this series to get overall picture of data management.